Schizophrenia is a personality disorder that leads subjects to have psychotic behavior. The most common notion of schizophrenia are people with visual and auditory hallucinations, which is true for many patients, but it is never the same for different individuals. There are many types of schizophrenia, and along with it, many positive and negative symptoms. Positive symptoms may include hallucinations, paranoia, and abnormal motor actions, such as catatonia. Negative symptoms may include lack of speech and poor cognitive behavior, anhedonia, which is the lack of pleasure, disorganized thoughts and speech, and overall impairment in the ability to communicate. There are many theories as to why people develop schizophrenia, and it fits in nicely with the nature versus nurture debate. It is accepted largely that schizophrenia is attributed to dopamine imbalance throughout the brain. But the reason why this imbalance occurs may vary from person to person. In every cell of our body, we have DNA, which is responsible for our genes, how we look, behave, develop, and the things we like or dislike. Our gene codes are responsible for different functions in our body. And the main genetic code researchers look into when researching schizophrenia is the gene COMT. COMT is responsible for dopamine metabolism in our bodies. During gene copying, it is rare that the body ever makes a mistake, but when it does, specific gene codons are copied the wrong way, and they are inheritable, and may or may not make a functional difference. These mistakes are called SNPs, single nucleotide polymorphisms. When it comes to COMT, alleles, or different versions of the same gene, may change the release of dopamine, or number of receptors there are in an individual's dopaminergic pathways, to which there are 4. The nigrostriatal, mesolimbic, mesocortical, and tuberofundibular pathways. Different alleles of the COMT gene affect everyone differently. They may increase the release of dopamine in the substantia nigra, an area of the brain dense with dopamine neurons, causing positive symptoms to rise, or it could reduce the number of receptors in a certain area of the brain, such as the mesocortical pathway, leading to negative symptoms to be more prevalent. Another factor that may affect the COMT gene, or any gene for that matter, are epigenes. Epigenes are chemical tags on histones, which is the DNA's protein, that cause the DNA strands to wrap themselves tighter or more loosely around the protein. This makes certain strains of genes more or less accessible. An epigene usually responsible for suppressing certain genes, making them less accessible to an enzyme, is methyl. Genetic methylation inhibits gene expression. Epigenes are known to be caused by outside factors, such as pollution, prenatal pathogens, or stress. Mothers that drink or smoke during pregnancy put their babies at a higher risk of developing schizophrenia. A condition called Rh incompatibility, where the mother's blood is different from the child's blood, also increases a child's chance to develop schizophrenia in the future. Stress may also be a cause of methylation, so much so that there is a near 50% rate of comorbidity or coexistence of schizophrenia and other stress disorders, such as generalized anxiety disorder or social anxiety disorder, agoraphobia, or PTSD, which are usually diagnosed a couple years before the schizophrenia diagnosis, which could be an epigenetic cause, but correlation does not prove causation. Other theories suggest that GABA dysfunction in the brain may also cause several symptoms of schizophrenia. It is thought that through methylation, or an allele of the gene, GABA production and reception is inhibited, taken away from one's ability to relax, and potentially causing paranoia. Studies on the psychedelic drug LSD also show an overactivity of serotonin in the brain, very similar to the ones presented in schizophrenic people. For treatment, antipsychotic drugs are administered, and their purpose is to antagonize dopamine receptors to stop all the positive symptoms of schizophrenia. First-generation typical antipsychotics such as haloperidol were successful in reducing hallucinations and delusions, but were notorious for their extrapyramidal effects. By antagonizing only D2 dopamine receptors, it would decrease dopamine reception through all the dopaminergic pathways, making negative symptoms more prominent. Decreased release of dopamine in the nigrostriatal pathway, and specifically in the substantia nigra, causes Parkinson's-like symptoms, stiff movement, and paralysis. This is called tardive dyskinesia. Second-generation atypical antipsychotic drugs, such as acetylpine, were successful in reducing these side effects by blocking specific serotonin receptors as well as D2 receptors, so neurons can still produce adequate dopamine amounts. The only exception is clozapine, only administered in more severe stages of psychosis under intensive monitoring, as it is known to damage the body's white blood cells. Psychotherapy, which is a mixture of cognitive behavioral therapy, personal therapy, social skills therapy, and group therapy, is to be administered. Compliance therapy also goes a long way in helping the treatment. Many patients decide not to take their meds, which completely undermine every treatment effort. Cognitive behavioral therapy is used to disillusion patients. Peripheral questioning is the method of trying to find gaps in patients' delusions to help make them realize what's real and what is not. CBT is also used to treat the comorbid disorders that may arise as a result of the schizophrenic life, or that may have been worsened if already present. CBT, cognitive behavioral therapy, also helps a person return to normal life and function to society's standards. One can never be cured of schizophrenia, but there are ways to control the symptoms. Hallucinations never stop. One can choose to make what they hear positive instead of a negative experience. With adequate and persistent treatment and coping, people can function normally again.